Welcome to Ms.Chiao's lecture videos. This is the unit on genetic mutations, and today we'll be covering the second video on eukaryotic gene regulation. Eukaryotic gene regulation, basically in eukaryotic gene regulation is very similar to prokaryotes, but there's more regulation factors because eukaryotic cells are more complex, therefore their regulation process is more complex. As a reminder, gene regulation is an on and off process, just like the picture here, demonstrated by CK12. There are basically switches in which your body determines which genes can be turned on to make certain proteins in which they need. Basically, all cells have the same amount of genes, same amount of DNA, the same kind of DNA. So your skin cell and your heart cell have the same kind of potential genes. The difference which makes them different has to do with kind of proteins in which it's differentiated from. So cells are differentiated based on which genes are turned on or turned off, which makes them different, a heart gene versus a skin cell gene. Now, the genes, and when you have gene regulation, related genes can be proximal or distal, which means when you're making a protein, in prokaryotic cells, the genes are close together. So most proteins are not just made from one sequence of genes. Most proteins are built by a composite of many different proteins put together to make one functioning protein. In prokaryotes, when they transcribe genes for each other, their genes are next to each other. But in eukaryotes, their genes can be next to each other and within the same 1,000 or 2,000 kilobytes away from each other, or that's proximal, which means it's close to each other, or distal, where it could be all the way on another chromosome to find a sequence of genes for another part of protein that's encoded for. So over here, before we move on, just as a reminder that there are regulatory proteins which land onto the part of the DNA, and that can help regulate whether it turn on or off. Of course, the presence of RNA polymerase to start the process to make, eventually you want to get an RNA transcript, which eventually will turn into a protein. Okay, so RNA, what are the different ways in which gene regulation is controlled? One of them is RNA splicing controls gene regulation. Just as a quick review, here we have a sequence of genes. This one's M7 and G, and over here it has exons and introns. Now remember, introns are the junk DNA that are excised out. When it goes through splicing, the sequence in which the exons bind together will determine what kind of protein is made. So in splicing, you can have 1, 2, 4, and 5, or you can have 1, 2, 3, and 5, depending either they're going to excise out the exon number 4 for this one, or excise out number 3 for this one. In this one, it will make a mature mRNA isoform 1, where exon 3 is skipped, and over here, exon 4 is skipped. So it becomes two different forms of protein. One will make a protein isoform 1, and here will make a protein isoform 2. Both with different functions. And of course, this eukaryotic cell is going to have your polyadenylation here. And this was taken from Bite Size Bio, today's website. Eukaryotes also have these composites called enhancers. Enhancers are the sites on DNA where loops to start an initiation complex. So basically, the DNA will loop itself as kind of like a homing device to have proteins start adding on to say, okay, we're going to start forming a complex to start the process of transcription. Eukaryotes also have an area where they start of the promoter, and it's called the Tata box. The reason it's called Tata box is because it has a specific gene sequence of TATAAA. Thymine, adenosine, thymine, adenine, adenine, adenine. So it would be Tata box. So an area of the promoter with the gene sequence Tata. It takes a number of proteins attached onto the Tata box in order for the RNA polymerase to bind onto RNA. So RNA polymerase just doesn't specifically go, oh, I can go and bind onto the promoter, which the area in which the RNA polymerase binds onto. It actually takes a couple other proteins to act like a signaling device to say, okay, now it's safe, it's okay to bind on. So you have an area of the promoter, which is an area of the gene sequence in which the RNA polymerase is supposed to bind onto. But before that, within that, they have a sequence of TAAA, where other proteins will attach to it before it binds on. So another way in which genes are regulated can be shown through developmental genes. An example of that are homobox genes. They regulate how things are developing, and this is very important in when species go through their embryo stage. So an example, there's Hox genes. They develop limbs and make sure they grow in the right place. So where your arms are at or where your legs are at, where that goes has a lot to where it's differentiated when it's small, and when there's certain mutation, things will go wrong. Over here we have an example of a fruit fly, and out of the fruit fly's head is a pair, and these are not antennas, this is a pair of a leg growing out of its head where the antenna should be at, but instead of antenna it has legs. So that's where things, really wrong things and mutations go wrong. Various factors in gene regulation. Well, before we go on to that, how does that apply to humans? Sometimes some people may have an extra appendage, an extra finger, that has to do with Hox genes too, or an extra toe, that will tend to happen as well. What are some other various factors in gene regulations? There are four main mechanisms. Regulatory proteins that bind on to the RNA polymerase. The enzyme itself can alter the shape of the RNA polymerase, so it will inhibit its ability to actually bind onto the DNA. So that could be one, and that could be an on and off switch. It may not be necessarily a mutation, something bad, but it's regulating, okay, we can't make any more on RNA polymerase, let's just go ahead and bind onto it, and then we will stop making that protein for that time being. Another type is activator proteins that help the RNA polymerase and the promoter. So over here, this is a really good example. Found this. Okay, so let's go look at these. These red proteins in this example are activators. They help the RNA polymerase and the promoter. Now, where is the promoter? You see this yellow box down here? This is the Tata box, and the promoter is a large area right here. The Tata box has a Tata binding protein. Now, the Tata binding protein has a whole slew of all these blue circles. These are different numbers that represent different co-activators, the kind of proteins that need to be there in order to have the activator bind on. And the activator bind on helps create this loop, this area here, to turn things on. Now, activator will help the RNA polymerase, and one of the things coming off of this Tata box are basal transcription factors. We're going to talk about that in a minute. There's also repressor proteins that bind onto non-coding regions that block the promoter regions. So one thing that helps is activators that help the RNA polymerase get situated and get started. There's a factor in which the repressor protein binds onto the non-coding regions. So over here, potentially, there's a repressor protein. It could bind onto a non-coding region, one of the junk DNAs that will start and block the promoter region. So here, a repressor protein could possibly block this area here. They have the right sequence to bind on and say, hey, RNA polymerase, you can't go anywhere. So that's another factor. And the last one are basal transcription factors that help park RNA polymerase, that helps tell RNA polymerase where you go. So in this huge complex of co-activators and activators, besides binding onto the TATA box, onto the TATA protein, of course first you have to get the TATA protein there, these round circles, B, and E, B, F, E, and H, these are basal transcription factors. Other proteins that need to bind on that which the RNA polymerase binds onto them and the DNA in order to start. So in eukaryotic gene regulations, the process in which it goes are very similar to prokaryote gene regulations. But you can see that there's a lot of factors that go into whether a gene regulation turns it on or off, whether it starts going or not. In the next lecture video, we're going to talk about what happens when things go wrong, what kind of mutations, chromosomal and single base mutations that happen, and what kind of diseases that we get for that. Until next time.